Chapter 4, Classification and Identification of Microbes. The learning objectives for this topic are: discuss the purposes of classification and identification of organisms, discuss the difficulties in defining species of microorganisms, list the hierarchy of taxa from general to specific, define binomial nomenclature, describe a few modifications of the Linnaean system of taxonomy, list and describe the 3 domains proposed by Carl Woese, and describe 5 procedures taxonomists use to identify and classify microorganisms. We're going to talk about Linnaeus and taxonomic categories. We're going to talk about domains, then taxonomic and identifying characteristics. First off, Linnaeus was a Swedish botanist that lived during the time when we had a whole lot of different taxonomic categories. In other words, lots of different ways of categorizing living organisms. In fact, with some new species like tomatoes and potatoes, sometimes they have 5 different names that were being used in the literature for the same species. So what Linnaeus did was develop a system of naming that could be standardized, and you could use one scientific name no matter what the common names were. So one of the things that he did was establish binomial nomenclature, so that every species had 2 names, a genus and a species name. And because we derived these names from Latin, we always italicize them. The genus name is always capitalized, the species name is always undercase. Sometimes we can use the first letter of the genus with a period and then have the species name to shorten things down. Well, this revolutionized the naming of organisms, and we use a variation of the Linnaeus taxonomic system. So let's talk about taxonomic categories as it was set up by Linnaeus. First, we have the kingdom. Linnaeus set up just 2 kingdoms, plant and animal. Then we go phylum, class, order, family, genus, and then species, with genus and species going into the name of the individual organism. Later on, a researcher by the name of Whitaker suggested that we change the kingdoms from 2 to 5. So we started out with just Plantae and Animalae, and we went to Plantae, Fungi, Animalia, Protista, and Prokaryote. So it kind of makes sense. Fungi are kind of weird beasties. They don't get up and move around like animals do, so they're like plants in that regard. But they don't photosynthesize, so they're like animals in that regard. They absorb nutrients, so in that way they're like plants. But once again, they don't photosynthesize. Also, they have chitin in their cell walls, whereas plants have cellulose. So, of course, they deserve their own kingdom. Protista, pretty much anything that didn't fit into plant, fungi, or animals that was still a eukaryote got tossed into Protista. And then we have anything that doesn't have a nucleus, anything that's not a eukaryotic cell. This system of 5 kingdoms worked for a long time. Then there was another researcher by the name of Woese who was working on identifying the relatedness of various bacteria, prokaryotes. And he was sequencing the small ribosomal rRNA DNA sequence. With some researchers have said that if there's a 3% deviation in the rRNA sequences, then it should be a new species. But he was working on this, and what he was working on were methanogens. They're anaerobic bacteria, meaning they don't grow in air, that produce methane gas in swamp mud. Yay, that sounds really exciting. But he made a really fascinating discovery. Ribosomal rRNA DNA sequences did not contain nucleotide sequences that were characteristic of bacteria. In fact, they were so different that it was approximately the same difference as between bacteria and eukaryotes. So he proposed that above kingdoms we add domains. And the proposed domains were archaea, to include methanogens, bacteria, and eukaryote. Since then, we have accepted these domains, and we go with them even today. Let's talk about taxonomic and identifying characteristics. But before we go into this, let's talk about the difficulties in determining species of prokaryotes. In eukaryotes, oftentimes what we use to determine a species is whether the individuals of that particular population can interbreed and cannot breed with related species populations. Well, that's all fine and wonderful with eukaryotes that can undergo sexual reproduction. Bacteria do not undergo sexual reproduction. They never interbreed within the population. They just make exact copies of themselves. When do you say that there's a new species? Well, let's talk about that. Here are the first 4 ways of identifying and characterizing bacteria. And we use these for many, many years. First off, physical characteristics. When we do a simple stain, we're looking at cell shape, which is a physical characteristic. Cell arrangements, we also use that as a physical characteristic because prokaryotes don't have arms and legs, and they don't have fur, and they don't have teeth, and we don't have other structures that we can take a look at. So we go with whatever we can. Speaking of arms and legs, we use flagella arrangement and placement, and whether it has a flagellum, whether it has a capsule, etc., etc. Now, once we run out of physical characteristics, then we go to biochemical tests. Whether the organism can ferment a particular carbohydrate, whether it ferments it with gas, without gas, can it break down certain amino acids, what are the byproducts of the synthesis, that kind of thing. Then we have serological tests. We take our handy-dandy antibodies that are specific for certain proteins, and we use them to identify bacteria. So once we've characterized, say, the causative agent of strep throat, we can get antibodies for that, and they will attach to the cells and cause them to clump. So if you go in with a sore throat and they do a rapid strep test, this is what they're doing. They're going back in the back and they're doing this kind of test with your throat swab. So a negative result would be the antibodies are not binding to the bacterium, or the archaea for that matter, and the sample does not clump. Where the antibodies are attaching and causing things to clump, you get this kind of result. Then we have phage typing. Phages are bacterial viruses, and they are very, very picky about their host cells. In fact, they are so picky that we can use them to characterize specific bacteria as to species and phage type. These were all fine and good, and they served us well for many, many years. But once we developed the ability to sequence nucleic acids, it opened up a whole new world. So let's talk about G plus C content. If you remember back to 201, you will remember that we have 4 types of nucleic acid, or we have 4 types of nucleotide bases. Here's a reminder, and we have adenines and cytosines and guanines and thymines. And in RNA we have uracil. Now remember that G plus C always hook up and T plus C always hook up. Well, we can use that to characterize bacteria. So we would take the G plus C content and divide that by the total nucleotide content, multiply by 100, and we get G plus C content. And G plus C content has been shown to be a reliable method of determining relatedness. It's a nice, quick, and dirty method of determining this. We also have improved upon that, and we're now able to sequence various DNA sequences, like going back to Woese and his research with the rRNA DNA sequences. So now we can not only take a look at GC content, we can also take a look at the similarity of sequences in certain genes that don't change very fast. Well, that's it for this topic. Here's the reminders of what the learning objectives are, and make sure that you come to class with any questions that you may have.